Hello and welcome to BiochemSerye, or the Biochemistry Lesson Series. This series is entitled The Basics of Chemistry. It is important that we review some basic concepts of chemistry before we dive straight onto studying the much more complicated world of biochemistry. This series is composed of 6 lessons. And this is episode 2, consisting of lessons 3 and 4. Please feel free to subscribe to be updated of the latest lesson installments for this series. This is the second episode of the Basics of Chemistry Lessons series and includes lessons 3 and 4. Please click on the card above if you want to see all the videos in the Basics of Chemistry playlist. Lesson 3 discusses the molecular structure, which includes discussions on bond lengths and angles, bond polarity, and bonds exemplified by hydrogen bonds. Lesson 4, on the other hand, delves on isomerism, the different types and conformation. The lesson also looks into optical isomerism, a concept that is very important in the study of biochemistry. The succeeding lessons are in the subsequent videos. Please subscribe and click on the notification bell to be notified of the succeeding lesson uploads. The physical and chemical behavior of molecules is largely determined by their constitution. That is, the type and number of the atoms they contain and their bonding. Structural formulas can therefore be used to predict not only the chemical reactivity of a molecule, but also its size and shape, and to some extent its conformation. Conformation, if we remember, is the spatial arrangement of the atoms within a molecule. In the traditional two-dimensional structural formulas, as we can see here, atoms are represented as letter symbols and electron pairs are shown as lines. Lines between two atomic symbols symbolize two bonding electrons, and all of the other lines represent free electron pairs, such as those that occur in oxygen and nitrogen atoms. Free electrons are usually not represented explicitly. Dashed or continuous circles or arcs are used to emphasize delocalized electrons. On the other hand, ball and stick models, as seen here, are used to illustrate the spatial structure of molecules. Atoms are represented as colored balls and bonds, including multiple bonds, as gray or white cylinders. The usual convention is gray represents the atom carbon, white for hydrogen, red for oxygen, and blue for nitrogen, although the relative bond lengths and angles correspond to actual conditions. The size at which the atoms are represented is obviously not to scale, as it is impossible to model. The third way to represent atoms, bonds, and molecules, as shown here, is the space-filling van der Waals models. These are useful for illustrating the actual shape and size of molecules. These models represent atoms as truncated balls. Their effective extent is determined by what is known as the van der Waals radius. This is calculated from the energetically most favorable distance between atoms that are not chemically bonded to one another. Moving on, let us now look into the concept of bond lengths and angles. Atomic radii and distances are now usually expressed in picometers, abbreviated as pm. To put everything into perspective, 1 picometer or pm is equal to 1 times 10 to the negative 12 meter, or simply stated, equal to 1 trillionth of a meter. The old angstrom unit, which is equivalent to 100 picometers, is not used anymore since it is now obsolete. The length of single bonds approximately corresponds to the sum of what are known as the covalent radii of the atoms involved. Double bonds are around 10–20% shorter than single bonds. In sp3 hybridized atoms, the angle between the individual bonds is approximately 110 degrees. In sp2 hybridized atoms, on the other hand, it is approximately 120 degrees. In molecular geometry, bond length or bond distance is defined as the average distance between nuclei of two bonded atoms in a molecule. It is a transferable property of a bond between atoms of fixed types, relatively independent of the rest of the molecule. In stable molecules, the attractive and repulsive forces are in balance, and thus bond lengths are therefore dependent on the bond strength, which is influenced by the number of bonds between the two atoms. As we can see here, the greater the number of bonds between atoms, for example, in double or triple bonds, the stronger the bond strength is, and consequently the shorter the bond length. Please pause the presentation and take the time to associate the relationships between bond strength and bond length. Let us now look into covalent bonds. A non-polar covalent bond is a type of chemical bond that is formed when electrons are shared equally between two atoms. This is termed as nonpolar because the difference in electronegativity is mostly negligible. It further means that there is no separation of charges between the two atoms, or both the atoms have similar electronegativity. And this type of covalent bond happens when the two atoms in the bond are either of the same atoms, or of different atoms with slight difference in electronegativities. To recall, electronegativity is a chemical property that describes the tendency of an atom or a functional group to attract electrons towards itself and thus the tendency to form negative ions. The nonpolar covalent bond existing between two different atoms is exemplified by the bond between carbon and hydrogen. Both the carbon and hydrogen atoms have similar electronegativities, and thus attract the shared electrons almost equally. On the other hand, let us now look into polar covalent bonds. Depending on the position of the element in the periodic table, as we have discussed in the previous lessons, atoms have different electronegativity values. Again, as a review, electronegativity is defined as the tendency to take up extra electrons. The values, shown here in the left image, are on a scale between 2 and 4. The higher the value, the more electronegative the atom, and the more it attracts electrons to itself. When two atoms with very different electronegativities are bound to one another, the bonding electrons are drawn toward the more electronegative atom, and the bond is polarized. The atoms involved then carry positive or negative partial charges. In the right image, the van der Waals surface is colored according to the different charge conditions: red for negative and blue for positive. Oxygen is the most strongly electronegative of the biochemically important elements, with CO double bonds being especially highly polar. To illustrate this further, when the atoms joined by a covalent bond belong to those with dissimilar electronegativities, the two atoms usually attract the shared electrons to different degrees, giving rise to a polar group or molecule. By definition, a polar structure, in the electrical sense, is one with positive charge concentrated toward one end, that is, the positive pole, and negative charge concentrated toward the other, or the negative pole. Compared with a carbon atom, for example, oxygen and nitrogen atoms attract electrons relatively strongly, whereas a hydrogen atom attracts electrons more weakly. Covalent bonds in which the electrons are shared unequally in this way are therefore known as polar covalent bonds. The best examples of polar covalent bonds are the bonds between oxygen and hydrogen, and between nitrogen and hydrogen. It is best to look at other bonds or interactions that are formed not by the sharing of electrons but by other means. In aqueous solutions, covalent bonds are 10 to 100 times stronger than the other attractive forces between atoms, allowing their connections to define the boundaries of one molecule from another. But much of biology depends on the specific binding of different molecules to each other. This binding is mediated by a group of non-covalent attractions that are individually quite weak, but whose bond energies can sum to create an effective force between two separate molecules. We will now be meeting the 4 different non-covalent bonds or forces that make intermolecular interactions optimally possible. These are ionic bonds, hydrogen bonds, van der Waals attraction, also known as London forces, and hydrophobic interactions. The first of the non-covalent interactions are ionic bonds. Ionic bonds are most likely to be formed by atoms that have just 1 or 2 electrons, in addition to a filled outer shell, or are just 1 or 2 electrons short of acquiring a filled outer shell. As animated here, an atom is transferred, for example, from sodium atom, which has a lone electron in its outermost shell, to the outermost shell of another atom, let's say chlorine, that is in turn lacking an electron. The sodium atom loses an electron and thus becomes a positively charged sodium ion or sodium cation. In turn, chlorine gained an electron and becomes an anion, a chloride ion. Because of their opposite charges, sodium cation and chloride anion are now attracted to each other and are thereby held together in an ionic bond. Let's now look at how water molecules affect the strength and stability of ionic bonds. Because of a favorable interaction between water molecules and ions, ionic bonds are greatly weakened by water. Thus many salts, including sodium chloride, are highly soluble in water, dissociating into individual ions such as sodium cation and chloride anion, each surrounded by a group of water molecules. This contrasts with covalent bonds whose strength are not affected by water in this way. The second non-covalent interaction is hydrogen bonds. What are hydrogen bonds? A hydrogen bond, often informally abbreviated as H-bond, is a partial intermolecular bonding interaction between a lone pair on an electron-rich donor atom, like nitrogen or oxygen, and the anti-bonding molecular orbital of a bond between hydrogen and a more electronegative atom or group. Or simply stated, this bond represents a special form of polar interaction in which an electropositive hydrogen atom is partially shared by two electronegative atoms. Unlike a typical electrostatic interaction, this bond is highly directional, being strongest when a straight line can be drawn between all three of the involved atoms. Do water molecules affect hydrogen bonding? Since water molecules are held together by hydrogen bonds themselves, it is expected that water weakens hydrogen bonds by forming competing hydrogen bond interactions with the involved molecules. It has been revealed that hydrogen bonds are only 1/4 its strength in water systems as compared to that of in vacuum. The third of the non-covalent interactions are what is known as the van der Waals attractions, also known as London dispersion forces. Let us look now into how van der Waals attractions come into being. Van der Waals interaction is a distance-dependent interaction between atoms or molecules. Unlike ionic or covalent bonds, these attractions do not result from a chemical electronic bond. They are comparatively weak and therefore more susceptible to disturbance. The van der Waals force quickly vanishes at longer distances between interacting molecules. To illustrate this further, the electron cloud around any nonpolar atom will fluctuate, producing a flickering dipole. Such dipoles will transiently induce an oppositely polarized flickering dipole in a nearby atom. This interaction generates an attraction between atoms that is very weak, but since many atoms can be simultaneously in contact when two surfaces fit closely, the net result is often significant. Please take note that these so-called van der Waals attractions are not weakened by water. The last of the non-covalent forces is that of the hydrophobic interactions. The hydrophobic effect is the observed tendency of nonpolar substances to aggregate in an aqueous solution and subsequently excluding water molecules. This force is caused by a pushing of nonpolar surfaces out of the hydrogen-bonded water network, where they would physically interfere with the highly favorable interactions among water molecules. The hydrophobic effect is responsible for the separation of a mixture of oil and water into its two components. It is also responsible for effects related to biology, including cell membrane and vesicle formation, protein folding, insertion of membrane proteins into the nonpolar lipid environment, and protein small molecule associations. Hence, the hydrophobic effect is essential to life, because bringing two nonpolar surfaces together reduces their contact with water. The force is a rather nonspecific one. This slide tabulates the effect of water on the strength of the different non-covalent forces or attractions. Take note which non-covalent bonds are mostly affected by water and which are not. Also, it is worthwhile to remember that in contrast to the effect of water on the non-covalent forces, water has absolutely no effect on the strength and stability of covalent bonds. Please feel free to pause the presentation in order to see and understand the effect of water on the different types of bonds. Let us now proceed to the next lesson. Isomerism. Isomers are molecules with the same composition. Or in other words, they share the same molecular formula, but with different chemical and physical properties. If isomers differ in the way in which their atoms are bonded in the molecule, they are described as structural isomers. Other forms of isomerism are based on different arrangements of the substituents of bonds or on the presence of chiral centers in the molecule. Let us now look at the different common types of isomers. Our first example are the cis-trans isomers. Double bonds are not freely rotatable, as we have previously discussed in the preceding lessons. If double-bonded atoms have different substituents, there are two possible orientations for these groups. For example, in fumaric acid, an intermediate of the tricarboxylic acid cycle, the carboxy groups lie on different sides of the double bond, or known as the trans or E position. In its isomer, malic acid, which is not produced in metabolic processes, the carboxy groups lie on the same side of the bond, or what is known as the cis or Z position. Cis-trans isomers, are also known as geometric isomers, have different chemical and physical properties. As we can see here, the melting points and the pKa values of the two isomers are markedly different. They can only be interconverted by chemical reactions. In lipid metabolism, cis-trans isomerism is particularly important. For example, double bonds in natural fatty acids usually have a cis configuration. By contrast, unsaturated intermediates of beta oxidation have a trans configuration. This makes the breakdown of unsaturated fatty acids more complicated. Light-induced cis-trans isomerization of retinol is of central importance in the visual cycle. Molecular forms that arise as a result of rotation around freely rotatable bonds are known as conformers. Even small molecules can have different conformations in solution. In the two conformations of succinic acid illustrated here, the atoms are arranged in a similar way to fumaric acid and malic acid. Both forms are possible, although conformation 1 is more favorable due to the greater distance between the COOH groups and therefore occurs more frequently. Biologically active macromolecules such as proteins or nucleic acids usually have well-defined native conformations, which are stabilized by interactions in the molecule, as we will see in succeeding lessons. The next type of isomers we will be discussing are the optical isomers. This type of isomerism arises when a molecule contains a chiral center or is chiral as a whole. Chirality, by the way, came from the Greek word chair, meaning hand. The most frequent cause of chiral behavior is the presence of an asymmetric carbon atom. That is, an atom with 4 different substituents. The presence of a chiral carbon leads to the appearance of structures that behave like image and mirror image, and that cannot be superimposed. As such, they are also known as mirror isomers. Then there are 2 forms, known as enantiomers with different configurations. Usually, the 2 enantiomers of a molecule are designated as L and D forms. Enantiomers have very similar chemical properties, but they rotate polarized light in opposite directions. Let's look at a particular example. The enantiomers of lactic acid. The dextro-rotatory L-lactic acid occurs in animal muscle and blood, while the D form produced by microorganisms is found in milk products. For example, the Fischer projection is often used to represent the formulas for chiral centers. While we are in the topic of isomerism, it is worthwhile to might as well look into a very important biochemical phenomenon dependent on isomerism: the Aconitase reaction. Enzymes usually function stereospecifically. In chiral substrates, they only accept one of the enantiomers, and the reaction products are usually also sterically uniform. Aconitate hydratase, or simply known as aconitase, catalyzes the conversion of citric acid into the constitution isomer isocitric acid. Although citric acid is not chiral, aconitase only forms one of the 4 possible isomeric forms of isocitric acid. That of the 2R, 3S-isocitric acid, the intermediate of the reaction. The unsaturated tricarboxylic acid aconitate only occurs in the cis form in the reaction. The trans form of aconitate is found as a constituent of certain plants. This concludes the 3rd and 4th lessons in the Basics of Chemistry lecture series. The next episode is comprised of the 5th and final lessons as shown here. Thank you very much. Thank you for watching this episode in this playlist. Check out the other videos in this playlist and please do subscribe to our channel and click the bell to be notified of new video episodes that will be regularly uploaded.